Okay. So, translation is one of the 4 steps that determine the abundances of proteins in a cell. And today, I'm going to describe quantitative models for many of the biochemical mechanisms that control translation rates. The mechanisms controlling translation can be divided into 2 major classes. One is controlled by, do I not tell? Oh, seems to have died on me. One is controlled by microRNAs and RNA binding proteins, which recognize cognate sequence motifs present in subsets of RNAs. These transacting regulators tend to be expressed in either a specific subset of cell types or to be only active under some physiological conditions and not others. So let's call these guys gene and condition-specific regulators. So the second mechanism class of mechanism is controlled by the general features, which include RNA secondary structure, upstream open reading frames, and codon usage. So the general features tend to be present in most or all mRNAs, and they tend to be acting in most or all cell types and conditions. So these 2 classes of mechanism are quite different from each other. We consider there to be 6 general features. I'm listing them here. We've developed linear multivariate models that predict control by these features quite effectively. We assign a score to each mRNA for each of the 6 features. As the models, as more features are added to the models, they become more predictive. So, for example, this scatter plot shows that a model using just upstream open reading frames and RNA folding is much less predictive than one that includes all 6 features. So in these scatter plots, this is the measured rate of translation determined from ribosome profiling data, and this is the predicted rate of translation based only on looking at the mRNA sequence. Our models show that the general features acting together control 70 to 80% of the variance in translation rates in these 2 yeasts, and something like 40 to 46% of the variance in Arabidopsis mouse and human. So this indicates that a much larger role for control by the general features than previous attempts to look at this had suggested. And here for each species, you're now just looking at the scatter plots comparing the prediction of the model to the measured data from ribosome profiling data. If you think about it, these results also set an upper limit on the amount of control by the gene and condition-specific regulators, the microRNAs, etc. This is simply given by the unexplained variance in our model, the gray shading. In addition, the fact that the general features account for a higher proportion of control in these 2 yeasts is actually consistent with the fact that these yeasts do not encode any microRNAs and they encode fewer sequence-specific RBPs than multicellular eukaryotes. So the differences in the heights of these blue bars make some sense. Now, to build models as effective as this, we actually had to spend a lot of time learning about how each feature regulates translation. I'm just going to have time to show you a little subset of that work for RNA folding. We used established computational methods to predict RNA structures and their Gibbs folding energy. These plots show the folding energy for a series of 35 nucleotide sliding windows across the 5' UTRs of 4 different genes. So each point along these traces represents the delta G folding energy for one particular window. There are regions along the RNA which are strongly folded, given by these troughs in the delta G values, and long unstructured regions such as this. To determine what aspects of these structures are actually the ones that are regulating translation, we did a number of things, including we took the delta G values for the most folded window from each transcript and made a feature of that. We made a separate feature out of the delta G values for the least folded window, and similarly for intermediate degrees of folding. What we see consistently across all 5 species is that the delta G values for the most folded windows correlate much more strongly with translation rates than those of the least folded window. And these are much better than you'll get if you just take the mean also. We did a lot more features and analyses and feature selection to get at the final model, which I'll skip over. But that result on the last slide shows you that the pre-initiation complex, remember, it binds to the 5' cap and it moves all the way down the 5' UTR till it hits the AUG and begins translation. The rate of that process is much more affected by hairpin loops than it is by unstructured regions. These most folded windows typically comprise a number of little folded regions. If you take the longest stem and you take the length of the longest stem that has paired nucleotides with no mismatches, that is much the strongest determinant of the overall translation rate shown by the light blue bars. But if there are additional paired nucleotides within these sort of 35 nucleotide most folded regions, perhaps another little stem loop over to the side, those also add a little more regulatory information. When we compare the length of the stems, the size of the loops, and other metrics, we can see they're remarkably similar across all these widely different eukaryotes. In addition, we've also found that the sequences controlling translation are similar across species. Here you can see that highly translated RNAs from S. cerevisiae and Arabidopsis are A-rich, the green, and G-poor, gold, in this region upstream of the AUG, whereas the mouse is very different, it's GC-rich. But if you instead look now at the changing frequencies in trinucleotides between highly translated and poorly translated mRNAs, you see, as you would expect, you get a good correlation for this metric between Arabidopsis and yeast with their similar A-rich sequences. But you also see a reasonable correlation between GC-rich mouse and Arabidopsis and yeast. So even though there's a very different GC content, the way that sequences are changing to direct translation rates are very similar across all the eukaryotes. Here I'm now showing you the relative contributions of each of the features to translational control. Between species, at least for some of these features, there are significant differences in the degree to which they exert control. But if you now look between tissues within a species, there's relatively little control differences in their contributions, or lesser, and this is shown for 3 tissues for mouse and 3 for Arabidopsis. Okay? And so this fits with the idea that the general features have a somewhat more constitutive role than the condition-specific control that is thought to be exerted by microRNAs. But what exactly is the role of the general features? Why on earth is this system bothering to modulate the rates of most genes in a somewhat constitutive manner, and why is it doing that using 6 different features? 6. So we actually have a theory. We actually think we have a pretty reasonable explanation for what's going on. We found that control is highly correlated between features. And the first part of the evidence for that is shown on this slide. Sorry. We found that the percent of the variance in translation rates that's explained by the sum of the contributions for each of the features for one of the data sets sum to more than 100%. So if you sum the percent contributions of each feature for this light blue data set, they'll obviously add to more than 100%. This one alone is 60%. And here we've more formally quantified the actually, I want to make sure that I said at the beginning that we have found that control is highly correlated among the features, and that's what that's the point I'm making at the moment. And so that was the first piece of evidence. But in addition, we found here we formally quantitated the correlation and control between pairs of features. And so this little red heat map here shows you that the degree of control by RNA folding in S. cerevisiae is highly correlated with control by upstream AUGs. The degree of their control correlation is about 60%. There are also, it turns out, within-feature correlations in control. Here, we've compared control by the most folded window within every 5' UTR with that of the second most folded window in every 5' UTR. And what these numbers tell you is that the second most folded window, its control is perfectly, 70% or more of its control is perfectly correlated with that of the most folded window. So it's collinearity. Okay? There must therefore be some powerful selective force that's ensuring that the sizes of stem loops in different parts of the 5' UTR are being ranked similarly. So in other words, strong folds are being ranked with strongish folds, weak folds with slightly weaker folds, as opposed to what we don't see, a lack of correlation. So this is what we don't see, a lack of correlation, uncorrelated, correlated, uncorrelated, correlated. This is what we see. So this kind of strong correlation between the features and within features will actually drive, it turns out, if you think about it, a relatively uniform density of particles along each mRNA. So in this case, you've got a high density of particles throughout the length of the mRNA because you have weak RNA structures, a lack of upstream ORFs, and efficient codon usage. Whereas down here, you have a low density throughout the mRNA because you have multiple strong stem loops, upstream ORFs, and inefficient codon usage. And let's think of a different situation. Let's say the arrangement had been different and you actually had one slow step on one gene that was rate limiting. And in this case, you get high densities here and low densities here. And if you think about these two setups, the total number of particles being needed to generate the same rate of translation is going to be higher here than in the previous case. So lots of particles needed, fewer, lots, but the same rates of translation, okay? It's known that the majority of ribosomal subunits are bound to mRNA in the cell. And ribosomes are limiting for cell growth. So in Drosophila, if you drop the concentration of any one of a whole bunch of ribosomal proteins just twofold, you'll slow the rate of cell growth enormously and slow the development of the embryo. Whoops. So this selective pressure will therefore force the efficient use of ribosomes in the cell. That's sort of a known thing. And I've just shown you the way to do that, and I think the only way to do that, is to have correlated control by all the features, okay? So that's what these things are doing. There's actually a second related advantage of keeping the density of particles relatively uniform along mRNAs. If there's a particularly slow step at one location in the RNA, literally the particles will rear-end I'm almost there. The particles will rear-end each other, and they will induce no-go mediated decay, and the RNA will be destroyed. And obviously, that's something the system needs to avoid. So the general features appear to have several purposes. One is the one you might think of, to change the amount of protein being made from each mRNA. But the second one, and I think perhaps more important, is they're just there to ensure that the system works efficiently. They're not really about abundances. They're about making the system work differently. So this work was a collaboration between myself and Jessica Lee, who's an assistant professor in statistics at UCLA. She's responsible for all the math. And Guolan Chu, who's at the Hutch. Thank you.
Hi, thank you for the very interesting talk. So I'm wondering, you observed that between some complex species, the control sequence of the mRNA are kind of inconsistent between species. So I'm wondering, between let's say Arabidopsis and the mouse data, is there any difference between their the difference beyond their sequence, like the modification on the RNA, and could that explain the difference between the species?
Okay, we haven't looked at modifications, so that's a simple point to make. But I think although there are differences like in total GC content, I think one of our aspects of our model is a motif finding thing where we naively ignore all knowledge of mechanisms and just say what motifs is changing. And that actually, it picks up a bunch of trinucleotide changes, but in the end, that's almost fully captures. It's basically all that's doing is just capturing RNA structure and upstream open reading frames. You can just show by looking at the collinearity between them that they're essentially getting the same information. So the sequence level, I would say that the surprising thing is not so much that they're different between the species, but there's more similarity than people would have thought. That's the thing I would emphasize at this stage. Despite very different GC contents, the way that things are changing is because it's RNA structure, and that's all it actually turns out the same. Certain dinucleotides like to be in loops, certain dinucleotides like to be in stems, and those things are coming through all the way through because it's just a very simple piece of biophysics that's true in all the cells. So I would more emphasize the similarity. And we haven't looked at the things that are going to be different, or all the microRNAs and stuff. Well, we're not looking at that. So that's where the difference, we've ignored the different bit. So that's why, hence the similarity.
Great talk, Mark. I have a quick question. So the hypothesis was that, in the end, was that there are multiple speed bumps at regular spacings, so that you don't have big traffic jams, right?
Yeah.
So how does that relate to the alternative solution of having one big speed bump at some place that achieves an overall similar reduction in the rate, or whatever rate you want to control it at, and not have that many bumps?
Yeah. I think an interesting parallel here is between this and metabolic flux analysis. So in the old days, there was the concept of the rate-limiting step for metabolic pathways. And Castor and Burns and others long ago showed that, in fact, flux is controlled, is delocalized throughout the system. And I think you have a similar situation. We have a flux of particles all the way down the system. And I think it's just, if you think of a rate-limiting step, it's just, you're just going to have a build, you'll just have things hitting each other. That's one thing. And I showed that a single rate-limiting step will need a total number of particles on the mRNA will be more, because you'll always have, because the low density is what the rate-limiting step is downstream of the rate system, there'll be a low density. By definition, there's going to be a high density above. If you had it more uniform, you would be more uniform over. Okay. You're using less particles.
It's less efficient then. Okay.
So I think all those efficiency arguments don't, people use the efficiency argument in biology all the time, doesn't really work. But in one case it does is ribosomes, because there's so many of them, and it's well known that their energy efficiency, use of protein,